Welcome back to Epistem, and today the topic of our discussion is glycolysis. The word glycolysis is composed of two words, glyco and lysis. Glyco means glucose and lysis means breakdown. So it is a process of breakdown of glucose. The aim of glycolysis is energy production in the form of ATP from the breakdown of glucose. And this energy production can be directly from the glycolysis process, or the end compounds, the end product compounds which are produced from the glycolysis, they can act as a substrate for the citric acid cycle and the oxidative phosphorylation. So in this way the energy is produced. This glycolysis process takes place in the cytosol of prokaryotes and eukaryotes, and it is one of the earliest metabolic pathway which is anaerobic in nature. The process of glycolysis can be divided into two phases, phase 1 and phase 2. So in phase 1 there is energy or ATP utilization, and in phase 2 there is ATP or energy generation. At first we will talk about the phase 1 of glycolysis. So in phase 1 this glucose is converted to glucose 6-phosphate by the hexokinase enzyme. And remember, when we have a kinase enzyme adds phosphate group to the product, and there is also involvement of ATP. So here the ATP, that is adenosine triphosphate, is converted to adenosine diphosphate. That means one phosphate group is released, because from tri we are converted into diphosphate. So the phosphate that is released gets added to the 6th carbon of glucose, and we get the glucose 6-phosphate. Next, phosphoglucose isomerase. Phosphoglucose isomerase converts the glucose 6-phosphate to fructose 6-phosphate. Now we all know that fructose is a structural isomer of glucose, that's why isomerase enzyme is used. Next, we have the phosphofructokinase. Again we have a kinase enzyme, that means the phosphate will be added. Now ATP is again converted to ADP, and the phosphate group is released, and this phosphate group will be added in the 1st carbon of fructose 6-phosphate, and it will be converted to fructose 1,6 biphosphate. Now this is the last 6 carbon compound, because after this 6 carbon compound will be broken to 3 carbon and 3 carbon compound, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And these two are isomers in nature, and it can be interconvertible by the triose phosphate isomerase enzyme. So ultimately in this process 2 ATP is utilized, that is at step 1 and at step 3. Now we will briefly discuss about the equilibrium of the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. So you saw that here we have two arrows, that means these compounds are interconvertible, and now we will discuss that in which conditions this will occur. So DHAP, or the dihydroxyacetone phosphate, it is a precursor of triglyceride and it is used for its synthesis. And glyceraldehyde 3-phosphate, it is an intermediate of the glycolysis process, because in phase 2 this compound will be converted ultimately to pyruvate. And so in order to favor the conversion of DHAP into G3P, that is the glyceraldehyde 3-phosphate, that is this conversion, and not the opposite, that is not like from glyceraldehyde 3-phosphate to DHAP, the cell must have a low glyceraldehyde 3-phosphate, because it is a simple idea that if we don't have much glyceraldehyde 3-phosphate, that means our body needs to produce them. So that's why there should be a deficit of this compound in our body. Now when we have a lot of ATP around our cell, that means our body is already having enough ATP to work. That's why body don't need to produce ATP, and that means the body don't need to perform the glycolysis process, and it again means that the body need not to produce the glyceraldehyde 3-phosphate, and as a result the DHAP concentration increases. And as the concentration of DHAP increases in our body, the triglyceride production is increased and they are stored as the fat. Now one way to shift this equilibrium to the right, that means towards this, that is glyceraldehyde 3-phosphate, is to create an ATP need. That means we need to make a ATP deficit environment in our body so that our body starts to produce more ATP. And more ATP production means the glycolysis process will be initiated and the glyceraldehyde 3-phosphate production will also be increased. And this ATP deficit or the ATP need condition is can be produced in our body by exercising. So when we exercise, our body needs more ATP, and so exercise utilizes the ATP, and this will pull the equilibrium to the right, that means towards the glyceraldehyde 3-phosphate production, and removing the DHAP and facilitating its conversion to glyceraldehyde 3-phosphate to proceed with the cellular respiration. So this is how the equilibrium is maintained. Now we will talk about the phase 2 of glycolysis. So here we have the glyceraldehyde 3-phosphate, and here glyceraldehyde phosphate group is added, and this addition of phosphate group is from this one, that is NAD plus, and phosphate group is present and NAD plus is converted to NADH, and this phosphate group will be added to this glyceraldehyde 3-phosphate and converted into 1,3-bisphosphoglycerate. So in 1 and 3 carbon numbers we have the phosphate group, that's why 1,3-bisphosphoglycerate. Next we have the phosphoglycerate kinase, again a kinase enzyme, and here what happens, the ADP, that is adenosine diphosphate, is converted to adenosine triphosphate. That is in this compound, in ADP, one phosphate is added, and this is this phosphate, that is the carbon number 1. The carbon number 1 on which the phosphate group is added, this phosphate will be now added to this ADP to convert it into ATP, and as a result the leftover compound will be 3-phosphoglycerate. So from 1,3-bisphosphoglycerate we are getting 3-phosphoglycerate. Next we have the phosphoglycerate mutase, which will convert 3-phosphoglycerate to 2-phosphoglycerate. Next enolase is there, and whenever enolase acts, the H2O is released and phosphoenol pyruvate is produced. And in the last step we have the pyruvate kinase, so again a kinase enzyme. So this kinase enzyme will add the phosphate group to the ADP and convert it into ATP. So this phosphate group will be added to this ADP and the leftover will be the pyruvate. Thus this is the end product compound of glycolysis. And here we see that we have produced 2 ATP in step number 7 and in step number 10, and total we get pyruvate, 2 ATP and NADH. These are the end products of glycolysis. Now we will discuss about the fate of pyruvate. So now pyruvate can now enter the citric acid cycle and the oxidative phosphorylation. So this pyruvate gets converted to acetyl-CoA, and this acetyl-CoA actually enters the citric acid cycle. Now whenever the ATP level is high in the cell, the rate of citric acid cycle decreases and the acetyl-CoA accumulates, because citric acid cycle is also energy producing cycle. So when ATP is high, so this the rate of this cycle also decreases and the acetyl-CoA accumulates, and this acetyl-CoA can be used for the fatty acid and the ketone body synthesis. So this is the pathway. So this acetyl-CoA is converted to fatty acid, alkenes, etc. Now we will talk about the fate of NADH. So we saw that in phase 2 this NAD plus is used and NADH is produced. So for the process of glycolysis to continue, we need the NAD plus, and this NADH can be converted to NAD plus again by reoxidation. Oxidation, we know that oxidation is the removal of the hydrogen. So this hydrogen is removed and NAD plus is produced, and this is done by the electron transport chain. So this is the process. And whenever we have a limited oxygen in our body, like whenever we are exercising in our muscles, we have very limited oxygen. So at that time the electron transport chain is not sufficient to convert the NAD plus to NADH to NAD plus. So in that condition, the NAD plus is again regenerated by the conversion of pyruvate into lactate by the lactate dehydrogenase enzyme. And when we have anaerobic conditions like in yeast, so here the pyruvate is converted to acetaldehyde by the pyruvate decarboxylase, and then this acetaldehyde again is converted to ethanol, and during this process NADH is converted to NAD plus. So this way the NAD plus is recovered in various conditions, like when there is anaerobic or limited oxygen and aerobic condition. Now we will talk about the energy yield of the glycolysis process. So net energy or net ATP gain in this process is 2 ATP. But when we deeply see this process, so what happens? Actually 4 ATPs are produced. Now it is because we saw that dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, these two compounds are produced in phase 1, and in order to continue the glycolysis we need the glyceraldehyde 3-phosphate. So one glyceraldehyde 3-phosphate is always present, and another one is produced by the conversion of dihydroxyacetone phosphate into this one. So the phase 2 occurs twice, and the phase 2 we all know that it is the process of ATP production. So from one cycle we are getting 2 ATP, and when the phase 2 occurs twice, so we are getting 4 ATP. So we are getting 4 ATP, and initially in phase 1 we utilized 2 ATP. So ultimately the net gain is 2 ATP. So this is all about glycolysis. Thank you for watching, and subscribe for more updates.